Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Bas van Steensel from the Netherlands Cancer Institute on this show. Please let me briefly introduce you to our audience. You received your PhD in 1995 from the University of Amsterdam. You then moved on to do a postdoc at the Rockefeller University and the Fred Hutchinson Cancer Center. And since 2002, you are research group leader at the NKI, and since 2014, you are head of division in gene regulation. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
I think just in high school, it helps to have a teacher that does a good job, being enthusiastic about biology. I was very good in high school at math and physics, sort of the hardcore science, and I naturally would have been natural for me to start to study physics or something like that. And I thought that's a little too straightforward. It would be more of a challenge for me to go into a slightly more soft science, and that would be biology. And together with that teacher that was making me enthusiastic about biology, even though I did not have the highest marks compared to physics and math, that made me decide to do biology. And then there was a brand new study at the University of Utrecht, which was called medical biology. And that became sort of a almost elite kind of training, because it was a very small group of people that were admitted. And so they gave an additional buzz to the excitement of doing this. And I was completely blown away by the first classes in molecular biology. I had not much clue about molecular biology, but I was so excited to read the whole Albert's book about molecular cell biology of the cell, that I basically spent most of my time not following the classes, but just going through the book, which meant that I actually failed my exam in molecular biology because I was not attending the classes and I was too much distracted by everything that was in the book. So it's kind of interesting that the only topic I ever failed an exam in is the topic that I'm now studying as a profession.
That's very interesting. But as you said, like physics and math are challenging, but I mean, molecular biology is challenging as well, right?
But in a different way. I guess for physics and math, you need a certain type of brain. It's more formula based, right? It's math. Math is formulas and strict logic, and biology is much more. Usually, you can't have a well-defined system that's completely behaving as predicted, and that made it more interesting and more challenging. I think
that's true. So let's talk about your work that centers around genome-nuclear lamina interactions, principles of gene regulation, and developing technologies for chromatin genomics. I want to start in the early days of your research career. There, you introduced DAM-ID, an alternative technique to map genome-wide occupancy of interaction sites in vivo.
Yeah.
Subsequently, you used this technique to characterize the part of the genome that is associated to the nuclear lamina, which you called LADs, so Lamina-Associated Domains.
Mm-hmm.
Could you talk about DAM-ID and how you use this technique to identify domains that attach to the nuclear lamina?
Yeah, I mean, the DAM-ID I developed when I was a postdoc in Steve Henikoff's lab, and Steve was incredibly generous and allowed me to use it as the basis of my own lab. And but that was also exactly the time when chip on arrays, chip was published by two famous labs, Rick Young and Pat Brown. They published it more or less the same time that we published the first DAM-ID paper. And so I had to think really carefully as a young lab, what is going to be the strength of my lab compared to them? And a big community. They published their papers in, I think, Nature, and so there was much more visibility, and you could see that chip and later ChIP-seq took off very quickly, and so we had to think carefully, what are our strengths? And with my lab, we defined first one strength, and that is you don't need antibodies for the proteins that we mapped with DAM-ID, because you just made a DAM fusion protein and that was it. So we started to focus on proteins that were difficult to chip. And that worked well for us. That worked well for us for a while. And then already when I was a postdoc in Steve Hennikov's lab, I thought about doing DAM-ID with Lamin. I made the construct, but I never had time to do it. And then
what was the motivation behind that? Sorry,
I just always been fascinated about, well, are there certain parts of the genome that interact with the lamina or with the nuclear envelope? That idea has been around already for a long time. And then in a collaboration with Maarten Voornerot, a colleague of mine at the NKI back then, we said, okay, well, the construct is already there. Let's give it a try. And in Drosophila cells, we saw already evidence for some domain-like structure. And then that was for us the incentive to do the same experiments in human cells. And then there was a spectacular domain-like structure. And that was super exciting. Yeah. And that made us also realize this is something that can be done with DAM-ID. That's difficult with chip. In the end, some people can do it with chip, but it's been much more difficult. And we were super excited about these domains, the LADs, and we
what are the properties of those LADs?
They're interesting because number one, they're really big, right? Hundreds of kb in size typically, that interact with the lamina, alternating with inter-LAD regions that are about the same size. And so you saw a very profound domain architecture of the genome. And then LADs, if they interact with the lamina, or LADs, genes inside LADs are expressed usually at very low levels. And that suggested that somehow LADs are repressive, which has been a long topic for us to pursue further to find out whether that's really true and whether interactions with the lamina are really important there. So it was, a lot of super interesting biology.
And what you also did is looking at the dynamics of LADs during processes like differentiation, and to visualize this process in molecular detail. So you generated high-resolution maps of genome-nuclear lamina interactions during subsequent differentiation of mouse embryonic stem cells via limited lineage-committed neural precursor cells into then terminal differentiated cells.
Yeah.
So what did you find there? And what made it interesting to look at the dynamics?
Yeah, what we saw back then was that so number one, for every differentiation step, you see lots of changes in these lamina interactions. Some regions go to the lamina, other regions go away from the lamina. And that pretty nicely correlated with changes in gene expression, right? So genes that went away from the lamina very often went up in expression, and genes that went to the lamina in the differentiation step went off. And even a little bit more than that, we had a 3-step or 2-step differentiation model where, as you said, we started with mouse ES cells, embryonic stem cells, and then converted them to neural precursor cells, and then from there on to astrocytes. And we even saw that some genes that moved away from the lamina in the first step of the differentiation didn't always go on in expression, but then that was predictive for that they could go on in the next step of differentiation, as if the detachment from the lamina preceded or maybe facilitated the activation in a later stage of development. So those were, exciting findings. Yeah.
This seems to be somehow comparable to those things that are now studied with ATAC-seq and RNA-seq, right? So that the chromatin first opens and then transcription may be switched on, maybe histone marks are put there. How does this then correlate? Is it that those ATAC-seq signals are only in the middle of the cells, or is this all interconnected? Probably.
Yeah, you don't. I think generally the ATAC-seq signals, you don't find them a lot in LADs. By all means, it looks like LADs are in sort of a heterochromatic state most of the places that are a repressive environment. So yes, you don't find a lot of ATAC-seq signals there. Yeah. Occasionally you do. There's some genes that escape the repressive environment, and there you do find, I'm pretty sure, I haven't looked at it carefully, but you would find signals there too.
So we are currently at the Amber meeting. I guess it's transcription and chromatin, or is it chromatin epigenetics?
It's transcription and chromatin, yeah.
And yesterday, and I'm not sure if this now plays into the LAD part of your work, but it was mentioned that you were looking at the 5 principal chromatin types, and you also defined those. And those are defined by unique yet overlapping combinations of proteins.
Yeah.
So how did you approach this? What did you find? And was this somehow a work that grew out of the LAT story, or was this independent?
It's somehow in parallel. As I mentioned, a strategy when we started the lab was that we would focus on, making maps of lots of proteins that were more difficult to chip. And I think we accumulated something like maps of 50 different proteins. And the whole idea was, okay, let's just take a real broad diversity of proteins, so that we get sort of an overall picture of which proteins like to go where. And then, this is by now quite a while ago, but when we did this, we found, we saw patterns. We saw sort of domains where certain combinations of proteins were going, other domains where other combinations of proteins were going. And back then we could convince ourselves, based on sort of statistical arguments, that we could identify 5 different types of domains based on protein composition. And back then we referred to them as chromatin colors. And that was a fun project. And I think since then, and also done by others, I'm not stuck to those 5 states. That was the first approach. And I think now it's almost a philosophical discussion whether it's a limited number of states or whether it's an infinite number of states, right?
Yeah.
You could, to some degree, you could say every locus has a different protein composition throughout the genome, so there will be 20,000 different states. Yeah, that's true too. But if you zoom out, there seems to be a limited number of states. And back then in Drosophila cells, we identified 5 states, and that helps a lot to think about conceptually, what does chromatin look like overall. We haven't really pursued that further. One of the states, what we call the black state, which was where there was very little gene activity. One of the markers there was the interactions with the lamina. And so to a large degree, black chromatin is actually the LADs. And we abandoned our work in Drosophila cells. We focused completely on human cells. And then we also said, well, the LADs are the most interesting part. And so we focused on that for a while at least.
Yeah. Thanks for coming back to the LADs, because that's where my next question would go. So it's always a question: do those domains stay at the lamina? Do they move? Are they like mobile? Do they come back after cell division? So what you did is you found or defined constitutive LADs. So how did you do that? What did you exactly look at, and what defines those constitutive LADs?
Yeah, constitutive LADs are by definition the LADs that, if you look in a lot of different cell types, those are the regions that seem to be always at the lamina. And I think we've done something like 10 different cell types, and something like 15% of the genome appears to be in these constitutive LADs. And what's interesting about these constitutive LADs is that they're really gene poor. Years ago, it was already noticed that there are parts of the genome that seem to be gene deserts. There's no gene, like megabase size regions that are gene deserts. If you now look back and combine this information, almost all gene deserts are constitutive LADs, and vice versa. I think it's a really interesting. So then the question is, why do you have these megabase size regions that have no genes?
I'm sorry to interrupt. Does this also include like stem cell like cells?
Yeah. I think yes. Yeah. I'm not totally sure whether we had that in that original data set, but no, I think so. Yeah.
Yeah.
But you can, there might be cell types where the whole thing is messed up. There's the retinal cells in nocturnal animals where things seem to be inside out. Also in senescent cells, you see that everything flips. So there might be exceptions to this rule. But in most cell types, you have these constitutive LADs. And I think they're really interesting because what do? There's no genes. So gene regulation seems to be not the major role. And they're interacting pretty tightly with the lamina. So maybe these things are there to give structure to the chromosomes in interface. To give overall structure, in addition to all the other mechanisms, loop extrusion, etc. This might be sort of anchoring points at the lamina that give most of the chromosomes overall structure. And that's not an easy hypothesis to test, by the way, because then you would have to delete all these large regions, and that's a horrible task. So we haven't done that.
So are those like spread along all chromosomes? Is there a pattern how they are spread?
Well, there's one or two small chromosomes that have only one or two of those. But yeah, all chromosomes have at least a few, and the big chromosomes have more. Mm-hmm. And so that sort of fits with that idea of giving chromosomes some structure. But as I said, it's not an easy one to prove or disprove.
So next, and this was done by Job Kind, who already was part of this podcast.
Oh, yeah.
You expanded the MID to the single-cell space by visualizing the fate of LUTs in single cells using a molecular contact memory approach. Maybe we can focus more on the biological results, as the technique itself was outlined by Job in episode 100. So what did you learn about LUTs by performing this single-cell experiments?
Yeah, so Job was able to visualize and even track the LUTs as a whole in living cells. And maybe one of the puzzling things is that these interactions of LUTs with the lamina are very dynamic. Even within one interface, I mean, they stay at the lamina, but parts of the LUT can move away about half a micron from the lamina and probably move back again. So it's sort of a turning and churning local kind of thing where the interactions are on and off. But the LUTs in one interface stay pretty much near the lamina. But then if a cell goes through mitosis, what we see is that roughly half of the LUTs nicely goes back to the lamina, but many of them don't make it back to the lamina in the daughter cell, which has changed our thinking about LUTs, because that means that LUTs are not regions that are always in every single cell at the lamina, but they have a certain probability of being at the lamina. And that raises questions about, okay, how important then is interactions of the LUT with the lamina for example, for repression of those genes? If they're not always at the lamina, then it's hard to imagine that contacts with the lamina play a major role in their repression. But there's other LUTs, and that's mostly these constitutive LUTs, that are going back to the lamina in almost every single cell. And so those are the stable anchoring point. But as I said, there's not many genes in there.
So where do they go? I mean, there is another compartment which is typically heterochromatin, which is at the nucleoli. Do they go there, or do they go just somewhere?
In part, they go to the nucleolus. We see that. You can visualize that by microscopy. You see that there's sort of a layer of heterochromatin around the nucleolus, and there seems to be indeed an exchange between lamina and nucleolus as the place for these regions to be. And several labs, including us, have put effort into mapping the nucleolus interactions, and you see that there's a partial overlap between NADs, nucleus-associated domains, and LUTs. And so, in part, there's sort of a dynamic exchange. Dynamic exchange, I mean, from cell cycle to cell cycle, not in the same interface, not within one interface. There are things after 1 hour into G1, they're pretty much settled and they stay where they are. But there might be some other chunks of heterochromatin floating around that are part-time at the lamina in some of the cells and not in others.
Another interesting thing that was mentioned already is that whether lamins are necessary for the LUT organization, and you looked at that. So do they really actively bind to the lamins, or what is the role of the lamins? Yeah,
it's still a bit of a field that's in flux, I think, but we mapped lamina interactions in cells lacking lamins or almost lacking lamins using another marker of the lamina, emerin, I believe. And we still saw the same kind of pattern of LUTs. And so, at least in mouse ES cells, it looks like lamins are not totally essential for lamina interactions. There was another paper by that has suggested that there is some effect of deleting the lamins, but I think these effects are very small. So lamins don't play a major role. It probably is some kind of really redundant mechanism where maybe there's other evidence that lamin A might play a role in another system, but redundantly with another protein called lamin B receptor. So it's probably a very redundant system that's not easily to mess up.
So did you look at cause or consequence? So what is the cause? So do they go there because they are inactive, or are they inactive because they are there?
Yeah, another tough nut to crack. What we do know, based on our data, but also others, Andy Belmont and Wendy Bigmore, if you force a gene to become active while it's at the lamina, it moves actually away from the lamina locally. The gene itself detaches from the lamina. So that is a bit of causality. But whether, and it's also not that being inactive as a gene is sufficient to go to the lamina, because there are many inactive genes that are not at the lamina. So it's not that simple. So what remains is whether maybe being at the lamina helps to repress those genes. So right, once a gene is active, it moves away from the lamina, and then maybe it stays away from the lamina and becomes more difficult to inactivate it. But suppose a gene is at the lamina, do the contacts with the lamina play a role? We have a paper in press in EMBO journal where we looked at this by doing a screen for proteins that are necessary for the repression of a gene inside the lad. And the screen worked really well. But what we find as proteins that mediate that repression or are involved in that repression, it's almost all chromatin proteins and no lamina proteins. Except maybe a little bit of lamin B1, but it's a very minor effect. And so, yeah, to me, that seems that maybe we've been thinking about this wrongly, and maybe the repression is really done mostly by chromatin components and not so much by lamina components.
Mm-hmm. Do you think they move away because there needs to be space for more factors to come in to activate transcription?
Yeah, it could be. How that movement away from the lamina upon activation happens, nobody really knows. It could be a simple competition game that there's now forces that pull it into the nuclear interior, transcription factories or whatever, and maybe that's how it works. Well, we don't know.
Okay, no. Yeah. So next, and this also goes into the realm of cut and tag a bit, maybe. You developed an antibody-based variant of the DAMID technology named PA-DAMID that allowed you to map and visualize genome-nuclear lamina interactions with high temporal resolution in order to investigate dynamics of LUTs during the interface. How does this work, and why do you think, or did you think it's necessary to develop this method, and what did you find using it?
Yeah, so indeed it is very related to cut and tag and cut and run. Right, we instead of a nuclease or a tagmentase, a TN5 tagmentase, we fused DAM to protein A, and hence you can now permeabilize cells, incubate with an antibody against lamin, and then come in with the protein A DAM fusion protein, and basically then the DAM is close to the lamina, and so it will methylate, adenine methylate the regions that are in touch with the lamina, and then you can map the adenine methylation pattern as we have always done with DAMID. So the nice thing of this is that we have a protein that we call methyl tracer, M6A tracer, that we can use to visualize in cells the location of that adenine methylation pattern that we lay down. So the cool thing is that before you actually go to a genome-wide mapping of the pattern, you can look in the cells. What do I see? And it's very comforting if you see a nice rim staining of the nucleus, and you know, okay, what I'm going to map from what I'm going to sequence is actually at the lamina. So that's a big plus. And sometimes you can use this method now too. You can also use it for other markers, like we've played with nucleolus markers, etc., and Ki67, another protein. It's very comforting if you see that the pattern that you map genome-wide is you can interpret it because you know where it's sitting. So that is it's a plus. But you cannot use it I wouldn't use it for transcription factors, for example. You will just see a signal throughout the nucleus and it's not going to help you. But for certain markers, particularly the lamina and other structural components, this is a useful tool.
Mm-hmm. Yeah, I want to go on to my next question, because you investigated the influence of CTCF, and this has been in my mind from the beginning, on the positioning and demarcation of LUTs, because CTCF has a big role in organization of TADs.
Yeah.
Right? And if you think of large domains, obviously what comes to your mind is TADs, because those are also like big domains that maybe play a role in gene regulation, or maybe don't. So what is the effect of CTCF on the LADs, and is it similar to TADs? How do the LADs compare to the TADs?
Right. So LADs and TADs, it's a bit complicated, but you could say to some degree that LADs are a subset of TADs, but it's not entirely. The maps significantly overlap. The patterns significantly overlap, but not completely. It's been always a bit puzzling. Sometimes a LAD can consist of multiple TADs, and maybe vice versa. And so there is an overlap. Already in 2008, when we published the map or the mapping of lamina interactions, we noticed that CTCF sites are demarcating very often the LADs. And so it's been on our mind for a long time. You would think that since CTCF is so clearly enriched at the borders of LADs, that they must do something there. So we have played with Degron approaches to deplete CTCF and remapped the lamina interactions. If I'm honest, the changes are very subtle. So the majority of LADs, many of LADs are marked by CTCF by sites at their border. If you deplete CTCF, a little bit changes. The borders shift a little bit, but it's not a spectacular effect. Mm-hmm. And that's the way it is. Again, and this is sort of a theme that has been a pain for us, is that LADs are incredibly robust. You can try to mess with whatever protein you like, and they laugh at you, and they just stick where they are, which indicates that it's a very... these are enormous domains, and if you think about evolutionary, how they have evolved, probably it's lots of mechanisms that each contributed a little bit to the interactions with the lamina or keeping it away from the lamina. And there's not a magic bullet that you can knock out and say, okay, now the whole structure is messed up. And CTCF is one of them. It's not a magic bullet. It contributes a little bit. That's what it is.
So they must be evolutionary conserved, right? So they must be found in other animals, or... Yeah,
LADs are very conserved. The positions of LADs in the genome are even conserved between mouse and human. You have to correct for, all the synteny changes that have occurred between mouse and human. But if you make a corrected map, the pattern of lamina interactions is very similar between mouse and human. We do find LADs in Drosophila. They're much smaller, but they're there, but gene spacing is also much smaller in Drosophila. There's something like LADs in C. elegans, at least the ends of the chromosomes, the more distal parts of the chromosomes tend to be at the lamina. So you seem to find it in many organisms, yeah.
So before we finish and transition into the outro, I briefly want to switch gears a little bit and talk about your efforts regarding transparency and outreach, if you also like.
Yeah.
On the one hand, you have an active blog where you post and write about your own research.
Mm-hmm.
So what motivated you to do this, and what do you write about?
Yeah. I think we as scientists, to be secretive and competitive is a silly game. It's also, I think, rather unethical, because almost everything what we do, including our own salaries, but also the funding for our research, what does it come from? It comes from taxpayers and it comes from charities, right? If you talk to people in the street and you say, oh, I'm competing with my colleague and I don't want to tell this person what I'm doing and we're trying to beat each other, then people will say, well, that's not why I gave you the money. You need to collaborate. And so I think it is unethical actually to compete with each other. We just need to be open and help each other. So for my lab, I formulated a couple of sort of transparency guidelines that we now stick to as a lab. And one of them is that, there's a couple of simple things. Everything we do, as soon as possible, we put it in bioRxiv, so it's out there before. One other thing is a part of transparency is for computer code. If you do coding for data analysis, now there's a lot of pressure in our field to put that out on GitHub for everybody. So if you publish a paper, But funnily enough, for the lab work, there's no such thing. There's no such thing of the record of all your experiments that you did. You only put on the figures, the polished figures, and maybe some supplementary figures, but the raw data, and some of the raw data, but not the process that brought you to it, right? All the failed experiments, whatever you call failed, are not there. And so we as a lab are also working to make our lab notebooks available online. That is not always an easy task, but it's more or less become standard policy of our lab now that once the paper is out or is about to be published, that we make it available either on the Open Science Framework or on Zenodo, whatever is easier, so that people can look at our notebooks and also see the failed experiments and see, sort of the side tracks that we had. And because if somebody wants to reproduce our results, then it might be nice if you only publish the beautiful experiments, but not the 10 steps to get there, then people may have to reinvent the wheel. And a notebook is not easy to read, but at least then people can look into the notebook and say, oh yeah, but you also had this problem, or yeah, you actually did something slightly different than I'm doing. So I think this is just and now with electronic notebooks, because our institute is completely went to electronic lab notebooks, and many institutes are. It's not so difficult to just click on a button and say export and make it available. So this is something that we're doing.
Yeah.
Those are some of the important aspects. And then there's the Open Science blog that we do. I encourage people in the lab to write about projects that are ongoing. So that if somebody else in the world is interested in this, that they can contact us way before we publish it. And we even have published about a project there that basically failed, and we put it out there, and the data are not of the quality that we could write a paper about it, but it's good to inform people. So we're hoping by putting that blog out that people can see what we're doing and contact us. I must say, it takes a bit of discipline in the lab to take time to write in the lab blog. That is actually limiting time. Everybody's so busy doing experiments and publishing their papers. So the frequency of the blogs is still not as high as I would like it to be. On the other hand, I don't want to force people in the lab to do it. So I try to encourage them. And so every now and then there is a lab member that says, okay, I have some time, I'll write something. Yeah,
oh, that's interesting.
Yeah,
I mean, I also have a personal blog, and it's always hard to find time and to write about stuff.
Yeah.
And even find. I mean, obviously you're doing stuff all the time in the lab, and you're busy. But what is it exactly what you want to write about, right? That's maybe also something that.
Yeah, exactly. And so we write mostly about sort of what the overall design or overall goal of the experiment and how we roughly approach it. And sometimes, depending on the person in the lab, there's an update. Okay, now I found this and now I found that. And so, time is the limiting factor here. Yeah.
So imagine you're due to submitting a grant proposal tomorrow. What would you have written into that?
Tomorrow? Well, yeah, I'm still excited. Okay, I'll tell. We have, it's on bioRxiv, we have put a lot of effort into making really good reporters for individual transcription factor activity. So we went through 86 different transcription factors and tried to design the best reporter for each of these transcription factors. It was a fun exercise because we learned a lot along the way about how transcription factors work and what are critical parameters for them. But we have now a collection of 60 or so really good reporters for 60 different transcription factors. So we can use this now in a multiplex manner. We can just take a dish of cells and measure the activity of all those 60 transcription factors in one go. It's a pretty straightforward experiment. And so I would love to do this now to study, I'm in a cancer institute, so to study some well-known cancer signaling pathways such as EGF receptor signaling or just a whole bunch of other signaling, of which are in detail described what's happening in the cytoplasm. The whole signaling cascades are known. But then, if you look at all the cartoons that are online that describe these pathways, then there's a lot of detail in the cytoplasm, and then there's an arrow into the nucleus, and that says proliferation or something like that. And sometimes a few transcription factors are known, but barely characterized. So I would like to now figure out what happens in the nucleus, which transcription factors are actually controlled by these famous cancer pathways. Mm-hmm. And so that's a project that we're just booting up in the lab, and I haven't written the grant about it, so it's about time.
Okay. So for the last 35 minutes, we have been on a journey through your scientific career. Obviously, we were not able to touch all of your work, and I only focused on the lab work. However, did we miss something important, or would you like to add something?
No, well, the general philosophy of our lab has been that you can make progress by developing new technologies, and you mentioned the DAM-ID and all the spin-offs of that, but we've also put a lot of effort into. One thing is describing the epigenome and the spatial genome, etc. But what we really need now is perturbations of the genome. And of course, CRISPR editing is phenomenal. It's fantastic to make perturbations in the genome, but it's not so easy to scale it. It's right, making big rearrangements in the genome. Yeah, you have to do it almost one by one. Or inserting a reporter gene throughout the genome. Yeah, CRISPR is not the way to go. So we've put a lot of effort in the past decade or so in developing scalable methods to mess with the genome. One thing that we've put a lot of effort into is inserting reporters into thousands of locations in the genome so that you can start to study what is the effect of the chromatin context on gene expression. Yesterday at this meeting, I presented work where we now also locally can insert a reporter in about a region of a megabase into more than 1,000 or 2,000 locations, so that you can really finely map how the activity of a gene depends on its precise position in the genome. And this kind of perturbations is what we need. We need to mess with the genome to understand how it works. And so we need to move from description to causality. Yeah. And it's not an easy one. And it needs to be scalable because the genome is way too big. And that's what my lab is mostly focusing right now on building those tools and then having fun biology with it. Yeah.
So to finish, what is your personal definition of epigenetics?
Well, I'd like to stick to the definition of heritable changes in gene expression. That's what I like. Heritable, at least across mitosis. And then I also think that histomarks and DNA methylation play a role, but don't forget transcription factors. Transcription factors are beautiful epigenetic mechanisms. Think of I think it's the Lac repressor even, right? There's all these autoregulatory feedback loops of transcription factor. The transcription factor, suppose the transcription factor switches on its own gene. That's a beautiful epigenetic system because once the factor is active, it will keep its own gene on. And then that's also transmitted to mitosis because the transcription factor will just be distributed over the two daughter cells and again, keep on the gene. So it's a single gene system. One transcription factor regulating its own gene. That's a beautiful epigenetic system. It's been found in bacteria, but it's also, in a way, the Yamanaka factors are also an example of that, right? It's not one factor, it's a few. But together, they form a beautiful epigenetic system. So my favorite epigenetic systems are actually transcription factors. And I'm saying this, of course, to provoke you a little bit.
Why? I mean, I also like transcription factors, and I think they should be a part of the definition.
Yes. No, so I think a functional definition is good. Biologists as a whole were very sloppy with definitions. That's in part because biology is hard to capture in definitions, because there's always exceptions to the definitions. But we're not very good at sticking to definitions. They evolve over time.
Everybody has his own experiences then. with their biology and their experiments.
Yeah, right. And sometimes definitions are hijacked by people who think they I want to be part of that hot topic, and then sort of redefine it a little bit to be part of the hot topic. And that's just what humans do, and scientists are humans, and so it's okay, but sometimes it makes things a bit more complicated.
Yeah. So thank you, Bas, for your time and for being on the show.
My pleasure.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.